Hi, what if I told you that biology is not just nature, it's code? And for billions of years, a microscopic word has been dragging in the shadows, creating the ultimate survival tool. Welcome back to Illusion Hashtag. Today we are moving past the theories and into the terminal. We are talking about CRISPR, the genetic cheat code that turned a bacterial defense system into the world's most powerful biological text editor. System lock initialized. It's time to hack the life itself. Let's get into it. Every system has its enemies. For billions of years, a silent cyber war has been raging on a global scale. The attackers, viruses, or phages. These guys are ultimate malware, continuously injecting malicious genetic code into the bacteria to hijack their internal hardware and turn them into virus factories. The impact zone is massive, affecting roughly 90% of archaea and 40% of eubacteria. We are talking about an urgent system alert on the planetary level. Biological software is being compromised every single second. If bacteria did not find a way to patch these vulnerabilities, life as we know it would have crashed a long time ago. They needed a firewall and they built a legendary one. Enter the ultimate adaptive firewall. To survive this onslaught, microbes evolved a literally immune operating system, CRISPR and Cas. But this is not just a basic block list. It's an adaptive system that actually remembers its attackers. Think of it as a two-part security suite. CRISPR, which is the memory bank, it stands for Clustered Regulatory Interspaced Short Palindromic Repeats. Basically a database where the cell stores snippets of enemy code for future reference, and Cas represent the executioners. These are CRISPR associated proteins, nucleases, helicases, and polymerases that act as the system's active security drones ready to hunt and destroy anything that matches the database. It's the perfect synergy of data and destruction. They don't just survive the attack, they learn from it. To understand CRISPR, you have to change your perspective. Think of a cell as a biological computer. Your DNA, that's the execution binary, the source code that tells the system what to do. The ribosome acts as a compile engine, translating the code into proteins, which are the functional output module of the cell. But every coder knows where there is a code, there is a bug. There's where the CRISPR-Cas9 comes in. In the architecture map, it's a debugger and editor. It's a tool that allows us to go into the file path of life and find the specific line of code and rewrite in real time. System status online. We are moving from observing the software to actually programming it. To catch a killer, you need a mugshot. In the bacterial world, the mugshot is stored right here in the CRISPR array. Let's break out this anatomy of the biological database. First, you have the leader sequence. Think of it as the start button. It's a conserve upstream codon that triggers the transcription of the entire system. Then comes the repeats. These are identical checkpoints, usually 23 to 47 base pairs long, that act as the structural framework of the memory bank. But the real stars are the spacers. These are 21 to 72 base pairs snippets of actual alien DNA stolen from previous invaders. This is hyper-variable viral memory. Each spacer is a unique record of past attack, allowing the cell to recognize that specific virus if it ever tries to breach the system again. It's literally a library of enemies, and we are about to see the cell knows exactly how to use these files to its advantage. This is not just a one-off defense, it is a full cycle biological operating system. This loop has been the standard operating procedure for bacteria for eons, and it runs in three clean phases. First, the system identifies a breach and snaps a piece of viral code, saves it in a hard drive. Next, it transcribes the saved memories into active guide molecules, essentially printing out the wanted poster. Finally, it deploys the security drones to find and destroy the target. This loop is why bacteria are the ultimate survivors. But here is the part that changed the science forever. What if I took this defense firewall and turned it into an offensive text editor? What if I could choose a target? Let's dive into phase one and see how that first snap actually happens. Phase one is the snap. Think of the Cas1 Cas2 complex as a high-speed digital camera. When a virus attempts a breach, this complex identifies a specific segment of the invader's DNA called a protospacer. It does not just look at it, it physically grabs it, snips it out, and saves it into the CRISPR array. This is the protospacer integration. By pasting this alien code directly into its own memory bank, the bacteria creates a permanent record of the thread. A new memory is saved. the database is updated and the system is now primed for the next time that specific virus shows its face. Let's look the system logs for this operation. It's a clean three-step process to build that permanent genetic memory. First, the system identifies a protospacer, a specific section of the invading viral double-stranded DNA. Next, the capture. The Cas1-Cas2 protein complex acts as a molecular seizure, physically snipping the protospacer out of the viral genome. Last, the storage. The viral snippets, usually between 21 to 72 base pairs, is then pasted into the bacteria's CRISPR array. It's become a new spacer perfectly sandwiched between highly conserved repeat sequence that are 23 to 47 base pairs long. The note taking is done. The cell now has a hard copy of the enemy's code saved into its drive. But a saved file is useless if you don't run the program. The mugshot has been taken. Now it's time to print the guide. Welcome to phase 2, print. Having the data saved is one thing, but now the cell needs to mobilize it. Think of this as the cell printing out its search warrants. First, the entire CRISPR locus or memory bank is run through a biological scanner in a process called transcription. This creates a massive continuous strands known as pcrRNA, but that strand is too clunky to use all at once. So, biological processor steps in and chops the long chain into smaller individual mature CRNAs. Each one carries exactly one specific viral mugshot. They are very active scout molecules. They are armed, they are precise, and they are ready to patrol the cellular perimeters for a match. The data is now officially live. Basically, having the virus's IP address is a start, but you won't stop the hack until you arm your security guards. Here is the technical process flow. First, read. It all starts with the leader sequence. The upstream signal acts as a trigger, telling the cell's machinery exactly how to start reading. Next, transcribe. The entire array is then copied into one massive strand of tcrRNA. This is the master list of every virus the cell has ever encountered. Last, process. Finally, the long strand is chopped down into individual mature cRNAs. Each one carries a specific viral mugshot, a custom guide ready for immediate deployment. The bounty is printed, the petrol is ready. Now we move to phase 3, seek and destroy. This is the end game, phase 3, seek and destroy, also known as interference. Now that the cell has printed its search warrants, it's time to send in the biological assassins. The matured CRN carrying that specific viral mugshot slots into the Cas nucleus. This nucleus is essentially a programmable weapon. It patrols the cell, scanning every piece of DNA it finds. It's looking for one thing, a perfect Watson-Crick base pair match. The moment the mugshot aligns with an invading DNA sequence, the trap is sprung. The Cas protein executes a precise double-strand break, cleaving the invader's genome into two. Let's zoom in on the tactical execution. This is not just a random collision, it is a highly coordinated strike. First arm, the mature cRNA, our genetic mugshot, binds with the Cas protein to form a ribonucleoprotein complex. This is the weapon, loaded and locked. Next, scan. This complex patrols the cell like a security drone. It's searching for any DNA that complements the base pairing of its guide RNA. Last, execute. Match confirmed. The moment the key feeds the lock, the Cas protein triggers endonucleolytic cleavage. It physically snaps the viral genome, neutralizes the threat instantly. Code broken. Target neutralized. Threat deleted. This is how bacteria have been winning the cyber war for billions of years, but we are just getting started. Wait, here's a critical question. If the Cas drone is programmed to hunt down viral sequences, why doesn't it even accidentally nuke cell's own CRISPR memory bank? After all, the mugshots are stored right there on the host drive. The answer is a mandatory authorization signal, the PAM, or protospacer adjacent motif. Think of it as a two-factor authentication code for your DNA. The Cas nucleus is smart. It won't even unzip a DNA strand to check for a match unless it detects these short hard-coded password sequences right next to the target. Viral DNA has PAM, but bacteria's own memory bank doesn't. This simple security checkpoint is how the system successfully distinguishes between self and alien code. No PAM, no access. Safe to say, nature cybersecurity is pretty airtight. Let's recap as how Cas9 work as a biological find command, but with a two-factor authentication. Factor one, the guide RNA, the search query. This provides a specific Watson-Crick base pairing needed to find the exact target. And factor two, the PAM sequence, the password. This is the crucial security check. This two-factor authentication system is what makes Cas9 so incredibly reliable. This is the ultimate combinations of a precise search query and a mandatory authorization signal. The microbial world has a massive diverse arsenal, but one specific class changed human size forever. Meet the MVP, Cas9. Nature does not just have one version of this tech, it has a whole diagnostic matrix of different routes depending on the mission. We are all about efficiency. Type 1, the heavy hitter. Honestly, too clunky for precision work. It uses a massive cascade complex and a Cas3 nucleases to trigger a full degradation cascade. It's powerful, but it's a lots of hardware to move around. Type 3, it's the specialist. Using CSM and CMR engines, it can target both RNA and DNA, but interestingly, it's spam independent, meaning it doesn't need the password we just talked about. Then there's type 2 Cas9. It is the chosen one because it is incredibly elegant. Minimalist build, maximum damage. This system runs on a single protein setup paired with an RNA duo, the crRNA and the tracker RNA helper. With RNase3 handling the backend, the engine is basically the flagship model of genetic engineering. It's simple. While Type 1 and Type 3 are cool, Type 2 is the sleek programmable drone and deadly accurate. This is the moment everything changed. Scientists realized something absolutely insane. This ancient bacterial firewall is completely programmable. We realized we did not just to wait for a bacteria to capture a virus to use this tool. We could perform a total system override. By swiping out that bacterial wanted poster with our own custom designed RNA guide, we can command this biological assassin to target, navigate to, and cut literally any sequence of DNA we want. We are talking about targeting human DNA, crop DNA, or any genetic sequence in existence. It's the ultimate cheat code. We have officially jailbroke nature's security system to use it as our own personal cursor for the genome. As we already explained, out of all the evolutionary variations, type 2 is the absolute goat. Let's look at the specs. Tracker RNA. This is the trans-activating RNA. Think of it as the precursor that base pairs with the repeats to help turn that raw pre-cRNA into a usable tool. Next, Cas9. This is the singular endonuclease powerhouse. It is the muscle that carries out the precise cut once the location is confirmed. By combining these elements, nature created a compact, highly mobile search and destroy unit. For us, this simplicity means we can easily drop this engine into almost any cell and expect it to run. The real genius of CRISPR was not inventing a new machine from scratch. It was the realization that nature's search and destroy drone could be hijacked. In nature, the system is a bacterial shield reacting to a viral RNA. But in a human context, we have turned that shield into a programmable cursor. By feeding Cas9 synthetic RNA coordinates, what we call single guide RNA, we can point this biological tool at any gene we choose. We have effectively decoupled the hardware from the software. The hardware, Cas9, stays the same, but we provide the software, the synthetic guide to tell to it exactly where to go. It is the ultimate biological exploit. We are no longer just observer of the evolution. We are the one holding the controller. When Cas9 reaches its target, it doesn't just tap it. It introduces a targeted lethal lesson to the cell's DNA. This is a double strand break or DSB. To the cell, this is a catastrophic hardware failure. It panics and immediately initiates emergency repair protocol to save its genome. But here's the secret. This panic is an opportunity. The cut itself is not the edit. The cut is just the open comment. This is exactly where we seize control of the code. We have broken the system on purpose, forcing the cells to open its source file so we can overwrite them. The cell is scrambling to fix the damage and it has two ways to do it. Let's look at the branching timelines of these repair pathways. Path A is the glitch, non-homologous end joining. Think of this as the backspace key. The cell hastily glues the broken end back together without a template. It's messy, leading to small insertions or deletions. We call these indels. This effectively knocks out or inactivates the target gene, great for breaking a bad line of code. Path B, patch, that is homology directed repair. This is the control V command. If we flood the system with a custom DNA template, the cell will use it to flawlessly repair the break. This results in a precise replacement, allowing us to generate custom mutations or insertion entirely a new function. We are not just breaking things, we are forcing the cell to fix them according to our design. To appreciate the CRISPR revolution, we have to look at the legacy tech. Before we had this programmable software, scientists are basically working with forged metal keys. We are talking about meganucleases, ZFNs, and talents. The mechanism was physical and brutal. They had to fuse DNA binding domains directly to Fock-I restriction enzymes. But here's the fatal flaw. These systems relies on complex protein DNA interaction. If you wanted to target a new genetic sequence, you could not just change a line of code. You had to engineer and clone an entire new custom protein from scratch. Let's look at why we call this era the dark age of gene editing. As we already said, to target a new gene, scientists could not just type a new coordinate. They had to physically engineer and clone a brand new complex protein from scratch every single time. It was a nightmare of trial and error. You are not just a geneticist, you had to be a master protein architect just to make a single cut. It was expensive, it was slow, and definitely was not scalable. CRISPR changed the game by moving us from custom hardware to programmable software. It is the difference between having to forge a new physical key for every single door you want to open versus having a master keycard that you can just reprogram on the fly. Let's look at the side by side comparison. This is the ultimate showdown. On one side, you have the old way, customs hardware. It relies on complex protein engineering where every single change in target requires a complete system overhaul. It's like having to rebuild your entire computer just to change one app. On the right, we have the CRISPR revolution, programmable software. Here, the sequence specifically comes entirely from simple Watson Crick base pairing. The hardware, the Cas9 machine, stays exactly the same. If you want to target a new gene, you don't rebuild the system. You just type a new guide RNA sequence. It's plug and play. It's the difference between a typewriter and a word processor. We have moved from the physical to digital revolution, and that is why we can now write the future of biology. We have moved from a long way from blindly hoping for random mutations. This is where we start writing the future. The potential impact zones are massive, ranging from medical to architecture. And the next update, DCAS9. Scientists have already enough the system to create deactivated Cas9. It is no longer cuts. Instead, it acts like a programmable Uber. We are not just editors anymore. We are architects. But even an architect needs to know how to jailbreak the system for maximum control. By mutating the Cas9 protein to remove its ability to cut, we have essentially achieved it. Meet the Cas9, a deactivated non-lethal version that acts as a programmable GPS tracker. Think of it as a delivery vehicle that can find any specific address in the genome without destroying the house when it gets there. This allows us to plug in various modules for a total system overhaul, such as transcriptional activators, repressors, even for epigenetic modifiers. Also, it can tag with fluorescence proteins to provide a live stream of the genome, allowing for direct imaging of live DNA in the real time. From an ancient battle war zone to the modern human laboratory, CRISPR has proven one thing: biology is not magic, it is code. For billions of years, this system was locked away, a private defense for the smallest organisms on Earth. But today, the terminal is open. We have moved from merely reading the book of life to actively rewriting the chapters. To wrap it all up, let's look at why CRISPR-Cas is the ultimate MVP of our biology hacking toolkit. We have moved from a simple microbial immune system to a globally programmable platform for precise genome editing. Here's a quick access summary. First, adaptations. The Cas1-Cas2 complex steal DNA snippets from the inheritors to build a permanent genetic memory. Next, expression. The stored memory is transcribed into RNA guides or custom search warrants to hunt specific targets. And last, interference. The crRNA Cas9 complex find the matching inverted DNA and snips it to bases. In the tech showdown, it's not even a fair fight. While the old guard relied on a high complexity labor intensive protein interaction, CRISPR used simple RNA DNA base pairing. It's low complexity, fast, and offer a massive scalability. It is the streamlined RNA guided goat of biology. plug and play programmability is no longer a dream, it's the current system status. And finally, we now hold the genetic cheat code. The only question left is how do we use it? If you are ready to keep decoding knowledge and defining reality, make sure you hit that subscribe button and join the Illusion hashtag community. We are scratching the surface of what this biological revolution can do. If you could write one custom line on RNA to edit a bug in the human operating system, what would you delete? Stay curious, stay hacking, and I'll see you in the next terminal.